This is the topic for this discussion session. It's you'll see in my presentation. I tried to make it a little bit provocative, just to probably stimulate the discussion. Of course, if you don't like it, just tell me, and don't be shy. And this pathobiome idea is actually take us back to what's the definition of a disease. If you go to Wikipedia or some other textbook, plant pathology textbook, they will tell you what the definition of the disease is a series of invisible or visible responses, like physiological processes of plant cells and tissues to a pathogenic organism or environmental factor that results in adverse changes in form, functional, or integrity of the plant and may lead to partial impairment or death of the plant parts or the entire plant. This is not mine. This was just taken from one of the books. This is the definition of a disease. So actually, we all agree that the disease is a dynamic process. It's not a static. It's not an object. This is the same idea I tried to present this morning. And we all learned Koch's postulate, and this is actually advocating the single pathogen, one disease concept. You have a disease, you isolate the pathogen, purify it, and then you take it, inoculate the plant, you get the same symptoms. So this is the cause of the disease. Well, yeah, of course. I'm not claiming that disease is not the pathogen is not responsible for a disease, but this is the concept that we all know and we all adopted, and of course we use. Now, if we look on post-harvest, so in post-harvest we have different pathogens with different biologies. We have the necrotrophics and the hemibiotrophics, wound pathogens, non-wound pathogens. We all know this biology of pathogens. And we have also, in post-harvest, we have diseases with a multi-species causal agents. It's mixed infections or a disease complex. In many, in some cases, we see a lot that disease is caused by a complex of different pathogens. And if you try to isolate one and then infect it, you will not get the same disease. And sometimes it's really puzzling what's really going on. And we all know that. Of course, now we know that about this metagenomic. In the metagenomic era, we know that, of course, we have so many other microorganisms that are inhibiting or colonizing fruit. Most of them, the vast majority of them, they don't do anything, and there's a huge amount of work in the literature. I'm not going to explain to you in details what's going on, but exploring the microbiome at the holobiont concept is gaining momentum in recent years, and so many works. You go and have a look, make a search, and you will find hundreds of different publications about this subject, and even an endophytes, as an endophytes. There's an article by Gabi Berg group showing that in one apple you have like 1 million or more bacteria as an endophytes. So we know that microorganisms are symbionts on fruits, vegetables, whatever, and we can find them everywhere. And when we look on the level of the metagenomic level, we see a strong cross-kingdom correlations between bacteria and fungal community. They always interact, and you see this is actual analysis we did in the past, and you see a very strong correlation with the cross-kingdom interactions. So there are a lot of communications going on between these microorganisms, and if we'd like to understand what's really the disease or the pathobiome, we need to look at the pathogen in a more wider microbial community. It's not only at a single pathogen. We'll just look where this pathogen is, in which environment it is, and what are the interactions that are taking place. And if you look here, we can analyze what's the interactions between the different microorganisms on the fruit. We have exclusion, co-presence, and fungi and bacteria behave as a social groups. As a social groups via cell-to-cell communications, and they always in the dynamic interactions between all components of this. So and then I explained this thing this morning. So we are moving from this disease triangle to a more complex environment. It could be a pyramid. It could be different things. Host, the environment, host plant, and the symbiome. The symbiome. This is a collection of the different microorganisms that are there. They're not doing anything. They don't harm the fruit, but in a certain conditions, the disease will develop. We don't know still what's the interaction that leads to the development of the disease. Okay, so and this is really this is my talk, and a microbe plus virulence factors equal a disease. There are so many applications now that actually question this. We have a pathogen equipped with a virulence factors, whatever, so many, and this equal a disease is really questionable. This is not my invention. This is in the literature. They start questioning this, and the accompanying organisms are likely to mitigate or enhance or facilitate or enable, whatever we would like to call it, the pathogenic effect, and should thus be considered as part of the disease process. This is where the pathobiome concept comes. We look at it in a holistic or a holobiont concept or a holistic approach. So all this complex of microorganisms that are involved in the disease process, this is the pathobiome, and they most likely that interact with each other to facilitate or enable the diseases. This is the idea. And I was looking in the literature, and this is amazingly you can see that actually fungi that were the pathogen that we are dealing with, they have endophytes. They have bacterial endophytes. This is, of course, from published data. So look at this hyphae. These all have endophytic bacteria, and this is and they could be also exo and endo bacteria or microorganisms that are associated with fungi, and what's the impact of these endophytes on fungal fitness and regulation, modulation of pathogenicity factors. There are some work to show that, for example, there is Rhizopus, I think. It's in Fusarium, right? It's in Fusarium. They have an endophytic bacteria. Once you remove the bacteria from the mycelium using antibiotics, the fungus stops to be pathogenic. So there's a lot of questions here, and now we lack. Right now, we lack comprehensive data about fungal microbiome, the fungal microbiome. I'm talking about the fruit microbiome, I'm talking about the fungal microbiome itself, and what's its role in the disease process. And I was really amazed to see this publication was in communications biology. And if you look, I don't know if you see the names, it's all post-harvest pathogens. You see the names of the fungi? They are all post-harvest pathogens, and all of them has bacterial endophytes. When you see something like that, that's really amazing. And after I saw this, we have some isolated Penicillium expansum in the lab, and we went and did some fish staining for bacteria, and you do find bacteria. You do find bacteria inside the mycelium of Penicillium expansum and Penicillium digitatum as an endophyte. So this raised a lot of questions. And to make it more interesting, if you go and have a deeper look, you will find endophytic bacteria in yeast cells. And we all use yeast for biocontrol. So this just makes you ask really a lot of questions. What's really going on, and ask so many questions. And as I see, more questions than answers. What's the role of the function of this endophytic wound colonizing bacteria in the infection? Not only the endophytic bacteria, but also the wound colonizing bacteria, does the pathogen co-evolve with endophytic bacteria in the fruit wounds itself? Maybe these pathogens or these fungi acquire this endophytic bacteria from the wound itself. Once they start the infection, they just acquire the bacteria, and then some communication is going on between the bacteria and the fungus, and this on the level of regulation of pathogenicity. So, this is an open question. And do endophytic wound colonizing bacteria have a role in the biocontrol system itself? Once you apply our biocontrol system into the wounds, and all of a sudden these wounds are heavily colonized with bacteria. What's going on there with our biocontrol and the bacteria itself? They promote or inhibit biocontrol activity. Okay, so the wound. What's the definition of the wound? How we define a wound? Okay, so the wound or injury is defined as a natural or structural or artificial, could be mechanical opening that serve damaged tissue, right? And then a bruising, is it a wound or not a wound? CI, cold injuries, is it also, this is a wound or not? Or a chemical injury, I don't know. Infections through wounds actually represent the vast majority of post-harvest diseases. And only part of the wounds and the fruit will be infected. Not all the wounds. If you have a wounds on the fruit, look at them. Not all of them will get infected. Why? Only part of them will get infected. And of course, resistance will develop. And I just was collecting different types of the wounds. Look here, is the lenticel is a wound or not? This is an open. Can be considered a wound, or the calyx or the stem end opening. This is a wound, right? So we can look at this level. Okay, so let's assume that we are having a wound on the fruit. Look here, and we have an injury. Then in the wound we have a release of like volatiles, nutrients, sugars, etc. And then in the second stage, this is an information that the fungi sense. We have heard this morning about sensing, ethylene sensing, but these fungi are more smart. They sense even volatiles that are released from the wounds themselves. I'll show you now some results about that. So fungi sense that something wrong with the host. And then once they sense, they start germinate. And this is a work that we published many years ago in post-harvest biology and technology. Look here, this is a wound. This is a drop of Penicillium digitatum spores. Once we have a wound, we have a drop. The spores start germinating towards the wound. They have a directional growth to the wound. And at that time, we did study this, and we find that actually J proteins is the same sensing mechanism we hear this morning about ethylene. J proteins and GPRCs are involved in the sensing of, for example, limonene. Once you have an injury in citrus fruit, there's a a piece of limonene, they sense it. And we did that in vitro. And once you expose the spores to limonene, they will start germinate. So as you can see here, this is really very interesting systems to look at. And I went back to this same pictures. I wanted to have a deeper look on the wound itself. I never thought about it, but when I just plot this picture, this picture is taking 2008. It's so many years ago, and just you look on the wound. Look what's happening on the wound. They are heavily colonized by bacteria and probably some yeast. I never looked at this wound like that. I just wanted to have, just blow it, a little bit and see what's really going on inside the wound. Look what's going on the wound here. There are some yeast and bacteria colonizing the wound. Okay, so I said, well, let's have a look on the literature on some scanning electron microscopy of different lenticels or cracks or whatever. When you go there and you look deeply, you find this is a lenticel. See how many bacteria, it's colonized by bacteria. So all these cracks, all these wounds are not sterile. They are heavily colonized by microorganisms. And one experiment we did, very simple experiment. You take an orange fruit, you wound the fruit, inoculated with spores of penicillium. In the inoculation, in the inoculated wounds, then you go and count the number of CFUs, of bacterial CFUs. You have an increased number of CFUs, of bacterial CFUs in the wound itself, the inoculated wound, compared to the uninoculated. Of course, the uninoculated will also be colonized, but on the inoculated wound, you will have higher number of bacteria. So what's happening there? And this is my last slide. And when we looked more deeply on this wound, the inoculated wound versus the non-inoculated wound, we see a drastic differences in terms of alpha diversity. This is metagenomic analysis, what's going on inside the wound. Drastic, significant differences. Look at the alpha diversity. This is A and B. A is wounded with penicillium, and B just without. This is just the control, the non-inoculated control. You see the difference between A and D in terms of the diversity. And then if you look at the beta diversity, the composition, you will see that are totally different microbial communities in the wounds. So I think I will stop here. I'll just think about all these questions that I raised for the discussion. And about this pathobiome, most likely that these fungi, pathogens, whatever we call them, that make a disease, they are doing it with help with something else, which we still don't know, which is worth of studying in the future.
Okay, I think it's really late and it has been a very long day. So just what we have in mind is to raise some question. Maybe you have your own post-harvest disease.
Yep.
So I think it's worth to think and to exchange idea about what has been raised. I don't know if you want to intervene already, or?
Yeah, I have a question.
Sure.
So when you do these population studies, you're using qPCR?
No, it's metagenomics, metabarcoding.
You're not culturing. I'm getting at the point. Are they all alive, or are you just doing DNA amplifications of what's there? Because there are things that go up. Populations live and then they die, right?
Yeah.
So are all those different microbes that you've identified, do you know that they're alive?
Yeah, of course, yeah. This is one of the limitations of deep sequencing. It's just that you extract DNA and then you do this amplification, bacteria and fungi. Yeah, some of them will not be alive, but when you do culturing and CFUs, and then you see the high level of colonizing bacteria in the wounds, so they must, most of them must be alive. But of course, there are.
They might not all be the diversity that you're.
Yeah, but you know, I don't expect that, you know, the 90% of these microorganisms are dead. Part of them might be dead, but the wound is very rich environment, sugars and so many other things that is a very good environment for growth and colonization.
So that leads you to the next question. If
Yeah.
you have fungal pathogens there which have assimilation, so they're releasing enzymes into the environment which degrade the host cells.
Right.
The host cells leach out nutrients and the bacterial population explodes.
Right.
So they might not be directly involved with pathogenesis, but they might be a result of pathogenesis.
Could be. It could be, yeah, of course. You know, I just posed questions. Yeah, you know, this is, of course, this is a valid you know, assumption. But on the other hand, I can assume that maybe the pathogen is actively manipulating the microbial communities in the wound and promoting specific components of these microbial communities that help or enable the pathogens to infect the tissue itself. This is another direction that this could be.
Yeah, there are two questions.
Yeah.
Samir, so how do we expand this then to non-wound post-harvest pathogens? So if we're looking at something like Neofabria or
Yeah.
or so we're looking at a lenticel and early lenticel infections that then progress at much later stages. Are we saying there's similar signals that are going on there, or are we inferring that from the system? I don't know.
Yeah. Well, you know, I don't know me.
Yes,
I don't know either. But I just show you that lenticels are not really. This is a wound. This is not sterile, and it's heavily colonized. Once you have the fungus, you know, this is just open questions. You know, yeah, the fungus start probably germinating and colonizing the lenticels, but what else we have in the lenticels? We don't know.
But these hemibiotropic organisms might not go through that lemon, so they go right through the cuticle.
Yeah.
And then they survive as quiescent infections and then transform themselves to the pathogenic phase.
You're right. If you have an aprosoria like, you know, Colotroticum and others, they go darker. I'm not claiming that on all the system this is but this is a valid way of thinking. I'm not claiming that all the pathogens that we are dealing with should follow this type of thing. It should be probably different from system to system.
Well, that's the thing. There's a lot of different pathogens. And the Ramularia that you were talking about this morning seemed to go through lenticels.
Lenticels.
Yeah.
Yeah.
And so in that case, there might be this interaction.
Could be, yeah. Or it could be on the surface that is something promoting the germination to go in. I don't know.
There is a comment.
I also was thinking that the crucial step in the pathobiome is that it has to be validated experimentally.
Yeah?
Initially, when we plant pathology started, we had this coach postulates
Right.
using a single pathogen. But when now you introduce pathobiome, they also need to be validated experimentally.
Absolutely.
So,
Yeah, of course. I just, you know, I said from the beginning, I'm going to be provocative just to make people a little bit, you know, raise questions. Of course, all these have to be validated, of course, but we started to see that there are some truth for this direction. But of course, it has to be tested and validated.
Yeah, so surface sterilization doesn't always lead to perfect sterilize it, and you get 5 different things or 10 different things growing up.
Right?
So it's really hard to prove that the microbiome, if you can't sterilize 100%, how do you get to 0? In other
Yeah.
words, it's very difficult.
Yeah, of course. If you, for example, if you take citrus and wounds and you inoculate penicillium with antibiotics or with different things, with a fungicide that is not affecting, and see what's happening.
Well, if I can add a comment, you don't need to have a sterile fruit. I would say that all the microflora which is on the fruit at the end, it's also helpful. So we are discovering little by little that this microflora is working and living together with the fruit. So it is helpful. We don't need to eliminate.
Yeah,
Well, I was
this
thinking
is
this
the
comment
symbiome, yeah.
Yeah, yes.
Get to a 0 state and then put your inoculum on without any bacteria there.
It's not.
How can you do that?
Yeah.
Yeah.
Yeah, but
That's
if
true,
you can,
that's true.
for example, Jim, if you can find a bacteria or 2 bacteria, I don't know, and co-inoculate them with the fungus, and all of a sudden you get an enhanced infection. So most likely there's something going on. You don't need to be sterile.
I was thinking it the other way around, just putting the pathogen on a sterile
Yeah.
system to see if there's this enhancement
Yeah.
with the bacteria.
Libo Chang.
Listen here, you just talk the microbiome with the wound, but I think that the pathobiome may also related to the surface structure of the
Can you take the microphone, because probably the other people there, they don't hear you well.
The pathobiome I think it may also related to the surface structure of the fruit. This year we published a paper about the microbiome on the peach fruit, and we found the hair is related to the dynamic of microbiota.
Okay.
When we remove the hair, and the microbiota changed, and the disease, the postharvest disease happen is low.
Yeah.
Yeah. So
Yeah.
so
Yeah, this is a good point, of course.
On which fruit? Yes.
peach.
Peach.
Yeah.
Okay.
Because the peach has hair. Yeah.
Yeah.
Because then as soon as you remove that hair, of course you're creating a wound.
Yeah.
Yeah.
We use the brass. It's very soft.
Okay.
Mhm.
Yes.
Concerning cold injury that you referred to, I think that has nothing to do with pathobiome, since we all know that when we have abiotic stresses, that makes the tissues more vulnerable to pathogens. And I have to say as an example, Gnomoniopsis in nuts of Castanea sativa, which we thought that is an endophyte, and it is, but now we have a great problem with this, also in Greece.
You want to comment? Yeah, she talked about the effect of the abiotic in
Abiotic stress is
yes.
cold,
abiotic stress promote the pathogen.
cold injury or
Yes.
temperatures.
I think it is similar also in northern Italy with Pneummonopsis castane. Yeah, because it's the stressed plant that is becoming the right surface for the growth of the fungus, which is an endophyte normally.
If I'm not mistaken, the Americans have published recently in 2020 or 22 a paper that says that the main cause is the climate change, the high temperatures that we have the last years.
Yeah. Can I? Yeah.
It was always considered as a ubiquitous fungus, and now we have this problem. And it was the same for Ramularia colotini in barley for about 40 years ago, I suppose. So who knows what's this shift from a, let's say, saprophytic or ubiquitous fungus to a pathogenic one. So there seems to be going on something, but at the moment, we can't explain it.
Yeah.
Yeah.
Hi. I actually think this is a really interesting idea. I actually wanted to complicate it a little bit more. So in the beginning, he was showing the disease triangle, and instead of just having one host and one pathogen, having the host and symbiome in the microbiome of the pathogen as well. We could also think about the effects of other microbes within the environment that are also interacting with all of those microbes as well. I'm thinking particularly with VOCs that are coming from things that are not directly associated with either the host or the pathogen.
Yeah, good question. Anybody want to have a comment? You know, we don't want to have it like question answer. We'd like you to be involved. You know, we don't know many things. We're just raising this. So if anybody wants to comment to this, yeah, okay.
Yeah, thank you. I think at the microbial level, they are always not alone.
What again? Say
at the microbial level.
Yeah.
So when we view things at the microbial level, they are always not alone.
Okay.
For example, when you want to isolate a single fungus, usually if you use different media, it's possible for you to get some bacteria. But when you use selective media for the fungus, then you get what you are looking for. Also, the apple you viewed using the SEM, the same sample, if you view it with another equipment, it's most likely you see some other things.
Yeah.
Most likely nematodes or viruses. So what I believe is that most of these microbes don't usually occur alone. So that means we still need to look more deep into the pathobiome. Yeah, thank you.
Thank you.
Yeah.
Thank you. Yeah. Yes, please. Yeah.
I realize that you are looking at cross-kingdom interaction. So were you able, using this cross-kingdom network interaction, to be able to highlight those microbiome that are antagonistic and those that have potential to be beneficial?
Yeah.
Was it possible?
Yeah, yeah. If you do this analysis, network analysis, and microbial interactions, you can see which are the microorganisms that have positive interactions or negative interactions with whom. Yeah, of course, you can do that, but this is all computational models. And if you see something like that, you have to validate it experimentally. But of course, you can see this network of co-occurrence or antagonism. Yeah.
Okay, thank you.
Yeah.
Yeah, I can add.
Yeah, you can go on. Yeah.
Okay, maybe I can add just some points for the discussion. And just as in the case of Samir, it's only to give ideas.
Yeah.
Well, one is this. We have a lot of new disease and new reports. It means that every year. Well, there are more scientists, there are more tools, so this is one fact. But on the other case, I believe really there is an increase, and we have seen this already also in the plenary sessions of today. So this is something to be considered. And there was a comment this morning. I think there are different drivers that are helping this increase of diseases in every sector and in post-harvest. So one is the international trade, which is favoring. The pesticide use reduction. Europe is particularly affected, but also other parts of the world. The global warming and climate change, and the varieties. So all these aspects are increasing.
I would say that in the case of the rising of publications or reports, I would say that it has to do with the Chinese going into international publication.
Well, there are
First reports from China and Asia have been increasing a lot this last year. So probably it has to do also with that.
Yes, I would say it's more scientists, but also more problems at the end. It's not only more research. Okay, but just an example, in the case of citrus, I just add the citrus as an example. We have a lot of disease which can affect, for example, European production, and maybe not yet present. So the international trade could affect this introduction. About pesticide reduction, I just showed this example today, but I think that when you reduce the pesticides, you favor some saprotroph, epiphytic saprotroph. So maybe they were not really an issue in the past, and they will come out, and you don't know where they are coming from. And this is just another example. One is more American, but we also have it sometimes. Sooty blotch and fly speck. We also have it sometimes here in Italy. And it's really some saprotrophs that sometimes you cannot explain, you cannot reproduce very easily, but they are present. Or sooty molds. This is really something that is growing in some areas. We don't have it, but in other areas of Italy they are, for example, very strongly present. They are not pathogens, but they cause a lot of problems on the surface of the fruit. And well, we said maybe you try to eliminate one problem with resistant varieties, the varieties resistant to aposcab, but a new species of Venturia that has never been seen before becomes an issue. So you see that you try to solve a problem, but sometimes come out. Okay, this I could skip. And just this climate change, I think that we have alternaria everywhere. It's in our houses. We have people that are allergic to alternaria because it's present everywhere. We just monitored the rice field. 10% is alternaria in the rice. In orchard, 10% is alternaria. So alternaria is everywhere. But why it becomes pathogenic? Because it is growing together with the apple, the pears, etc. So yes, this is coming from yeah. Okay, that is just, and this is a paper which has been recently published by a Chinese group where they found what are the pathogen facilitators and antagonists in the establishment of the disease. So there are papers coming out to study what are the microorganisms that can help. They can be bacteria, they can be fungi. Well, this has been already faced before, so we don't have only wound pathogens, but hemibiotrophic pathogens that have first phase, then they are quiescent, and then they come out as necrotroph at the end. Okay, I just other ideas. Well, we have, as Samir has described, this dysbiosis. So the microbial equilibrium is changing due to several factors. Sometimes these are abiotic factors, but this is creating the conditions to favor the establishment of the disease. And this climate change is a major driver in creating this imbalance in the plant or in the fruit at the end. So I just give this as an example because we are studying this disease. It's a decline of fruit. We have lost something like 10,000 hectares in Italy in the last 10 years. And it's simply a very complex disease. And it's very complicated to understand what is the main cause of this. Because there is high temperature, which are favoring temperature of the soil. There is flooding, which is favoring. There is the soil structure, which is changing. There is anoxia in the roots, and there are pathogens. So just to answer to the question before, how do we reproduce the symptoms and the Koch postulates? You have to put all the elements together. So to reproduce the disease, we have to grow at high temperature with water and with the pathogen. So I think this is making really complex, these Koch postulates. Well, this is just what Samir said about interactions, microorganisms which are favoring or decreasing the disease. And I just add one more point of discussion. We said bacterial, fungal interaction are really important. Also, they say nowadays that maybe the hyphae of the fungi are the highways for the bacteria. So the bacteria are growing on the hyphae to penetrate, maybe because the hyphae are growing very quickly. The bacteria are slow, so they use the hyphae to penetrate into the fruit or the plant. And this is also something to consider. And I just add the viruses that we do not consider a lot, but the viruses, we have the phytoviruses, the viruses of the plant. We have the mycoviruses, so the viruses of the fungi, and bacteriophagy, so the viruses of the bacteria. Each one of these is changing the conditions. And just this paper, it's of 2023. For example, in this case, Botrytis cinerea, its virulence has been modified by the presence of a mycovirus. And already several papers demonstrated that the virulence is modified by the presence of these mycoviruses. So maybe we have also to consider sometimes this into the interaction. And that's all for
Okay, so let's, yeah, go on.
Concerning the case of kiwi decline, if I'm not mistaken, you referred to Phytophthora species as the agent and some Phytopithium. I have two questions. What is your opinion about Phytopithium? Because we seem to face them when we want to isolate Oomycetes. Now we found, we didn't know, we know Pythium and Phytophthora species. Now we have to face Phytopithium. What is your opinion about Phytopithium genus? And my second question is, if I'm not mistaken, they have found some bacteria causing decline in kiwi.
Yes.
So what is the main cause of kiwi decline? Do we know, or
No.
we are still trying to find it?
Well, you are from Greece, right? Yes. Is there also decline in Greece?
Yes.
Okay, because nothing come out from the literature. So in Italy, we think we are the only one with this issue, but it's in every region, and it's very strong.
I think so.
Yeah, just I say I don't have the answer because there have been so many hypotheses from all the scientific community. We don't know, but it's a plant that Italy was the first producer in the world, then the second after China, and now it's going quickly down. So we have cultivated this crop for 40 years, but now it's really dying. Suddenly, in the summer, the plant dies. And we don't know really why. But Phytoepithium is what is associated to the roots. The other species are present in the rhizosphere. But yeah, I think they are present, but also here the discussion is open. And about the bacterium, Clostridium, in my opinion, it's not involved, but well, some bacteriologists demonstrated that it could be also one of the agents. Yeah.
Thank you very much.
It's very complicated anyway.
Okay. Some more contributions? Okay. Yeah, I just wanted to ask what's your opinion about this fungal microbiome? Exo and endo microorganisms that we find in fungi. You know, in this roundtable discussion, we did not promise to give answers. Or just more questions and doubts and thoughts. Yeah, of course, yeah.
So Alternaria, was never a disease on almonds, and so a field disease on almonds in 1990, all of a sudden became a major disease on almonds, and we couldn't figure out why. We've grown almonds since early 1900s, so it was like, where did this disease come from? And so we started looking into this, and it was basically, we've now called it a disease of high production. And because they plant the trees closer now, they change the microclimate. Ubiquitous, we talked about Alternaria being ubiquitous, and if we plant trees further apart, the disease goes away. If we plant the trees high density, we put a lot of fertilization on the tree, we water more to get the trees to grow, we increase the humidity in the orchard, all of a sudden Alternaria has become, so we tell the growers, if you don't want to deal with Alternaria, just change the microclimate in your orchard, and it goes away. So this is a disease of high production, that's what we call it, of high inputs.
Yeah.
How do you explain that some Alternaria species are able to produce a highly efficient toxin, and others are not?
Yes.
And where is this linkage?
Well, all the Alternaria on almond has no toxin production. So it's so you're selecting for these toxigenic strains. This is the same in citrus. We have Alternaria on citrus, but it's not that there's no toxigenic ones in California. If we go to Florida, where there's high humidity and all that, we selected for the toxigenic strains causing fruit spots. So I think it's some of these new diseases might be from cultural practice changes in cultural practice. And so with high inputs, we get more diseases.
Yeah.
Anybody else?
Yeah.
You have comments on emerging diseases? Yeah.
Yeah.
Okay, I have two comments, two suggestions here. One is that when we talk about microbiome, when we do the biocontrol that, which means that we know we use one antagonist, for example, antagonist yeast, which can affect the microbiome of the fruits. However, if we use two of them, we use a complex antagonist yeast, maybe which can get the better biocontrol efficacy. My one suggestion is that when we research the biocontrol agents, we should use complex antagonist. This is my one suggestion. Another one is that, you know, when we do the control of the post-harvest disease, in fact, we don't know the mechanisms, especially in the molecular level. So we should do more research about the, for example, the fungi which can invade the fruits, the mechanisms in molecular level. For example, which genes take the action, which enzyme, for example, some fungi can produce some enzymes, which these enzymes can degrade the cell wall of the fruits. In fact, until now, we don't know clearly the mechanisms of the fungi pathogen which can infect the fruits. So my second suggestion is that we should investigate the mechanisms of the pathogen to infection the fruits, the molecular level. So this is my two suggestions.
Great. Thank you. Thank you very much.
Yes, there was.
First of all, very cool research, and I'm working on the microbiome impact over the conifer pathogen system, and I did some similar research as yours. I did the interkingdom and intra-kingdom microbiome interactions in this system, and from my research, I noticed that actually the interkingdom is much less than the intra-kingdom. So I was wondering, how about like in your research, did you also did the quantitative analysis of the interaction analysis of interkingdom and intra-kingdom, and how the situations in your research results? And the comment about my research experience is that even though the interkingdom interactions is very important, I think the intra-kingdom interactions you should never ignore, because from my study case, the intra-kingdom interactions is much more abundant than the inter-kingdom. And also, like, I also build these networks, and from the network mapping, and I noticed that some of the interkingdom, it's very conservative. The map is so conserved in a very small area. Yeah, that's my question.
Yeah. Which pathosystem you work with?
I'm working on the conifer Heterobasidium pathosystem. So Heterobasidium is a conifer root rot pathogen.
Okay. Yeah. This is very interesting comment. Yeah, about the intra and inter-kingdom interactions. Yeah, it's valid. Yeah, very good.
What about, like, in your research, do you also did this comparison between the results?
No. We have not looked at the intra at the intra-kingdom interactions. Yeah, this is very good comment that we need to think about.
Well, just my comment is that by studying this interaction between fungi and bacteria, I found more interest in the fungal interaction. And I believe there is much more competition there. Bacteria are, well, we skipped in some of our pathosystem at the end, but bacteria were a bit less important, at least this is what we have realized. So I think, yes, maybe your comment is correct, that intra-kingdom interaction are really strong. And just about the other comment, I think the synthetic communities or these communities of microorganisms could become really important in the future as soon as our research will be able to realize that not only one biocontrol agent could be applied, able to solve all the problems, but maybe 2, 3, 4, or even more microorganisms could be applied. I have an example from the grapevine where in order to enhance the capacity of the plant to resist to diseases, they are now creating synthetic communities, at least of 10 different microorganisms, each one with a different function. So this is also something that I think is really important.
Yeah, yeah. Actually, I can add to it. We have noticed that when we add, for example, biocontrol agents to the fruit, all of a sudden we see a very strong increase, for example, in bacillus. It's a natural microorganisms, and all of a sudden you see an increase in its counts, in its population, after you add specific microbial agent like yeast. And this makes you wonder why this bacteria, specific bacteria, is increasing while others are decreasing, like Pseudomonas and some other, Bentoia, for example. Bentoia, once you add a yeast, it just disappears. And all of a sudden you see an increase in bacillus. So these are the observations or findings that's worth thinking. And these interactions maybe there's in bioinformatics, or they call it guild. You know what guild is? Guild is just interactions between microorganisms that can help each other in type of metabolites. They exchange guilds, right? Guild. Yeah, this is a very expression. This is guilds. So now we have the tools to look at it in all these models, bioinformatic models, and test it in the lab. So there are a lot of things that we still don't know and worth thinking, and how we can manipulate the microbiome by maybe adding very simple compounds that will help specific component of this microflora to suppress the pathogens, or the other way around. When you, for example, add fertilizers, you might change the whole physiology of the plant and promoting something, Alternaria or some other microorganisms that help Alternaria to establish a high population or a high inoculum on the field itself. We don't know. Maybe this dense cultivation or high humidity is promoting some other components of the microbiome that we have never thought about. Yeah.
Yes, this is actually not a new phenomenon, because in the animal husbandry it's already well established that there is probiotics in rumen fermentation. So this is already well established concept. And then in agriculture, the concept of soil fertility that is mediated by some soil that's enriched with a certain group of microbes. So, but the issue is how we can identify this group of beneficial microbes so that it will be of relevance to plant pathosystems. Yeah, so this is the issue, because we know that they exist. There are some microviruses that without their presence, some plants cannot tolerate high temperature. So if you inoculate, expose those plants together with their fungi, then those plants can tolerate. So there are quite a number of cases. It's how do we apply the bioinformatics system to identify these beneficial microbes so that they will really have a positive impact. So,
yeah. Thank you.
Thank you. Comments? Yeah.
I feel so very sorry because I have many questions and not answers.
Yeah, we are in discussion, you know, just raising ideas for the future.
I'm thinking all this time about the case of Alternaria on malus, on apples. According to EFSA categorization, Alternaria alternata producing AM and AK toxin is listed as quarantine ones because they are non-European isolates. In your cases, have you examined? It's not that. It's not this case. Okay, I'm glad for that. Okay, Alternaria alternata non-European isolates producing AM and AT toxin. Okay, that's good. That's good news. Yes. And I have another question also concerning Ramularia and dry red cell rot. You have isolated Ramularia from this. Yes. It's not a non-parasitic disease. Okay, thank you.
Yeah, I just want to raise a question about the toxins, about fungal toxins. You know, you mentioned Alternaria, and you know, in the past, you know, I did my master's degree on Alternaria alternata, so I know a little bit about Alternaria, and you know, I'm asking, what's the role of this toxin? Is it pathogen? Is it essential for pathogenesis? Because there are so many different species that do not produce this toxin, and they are pathogenic. What's about patcholine in Penicillium expansum? Is it a pathogenicity factor or not? There are some strains of Penicillium expansum do not produce patholine, and they're still pathogenic. Yeah. We actually we did some work in the past, and we knocked out the gene for. It's actually one, you know, it's a whole pathway. It's all the genes involved in the pathogen production pathway, and the fungi is still pathogenic. So the question, what's the role of these toxins? I think that these fungi use these toxins to outcompete the other microorganisms that they are in their vicinity or their environment, in order to get an advantage and infect the fruit.
Well, that's how patcholine was discovered.
Yeah.
As an antibacterial, functional antibiotic.
Yeah.
Just after the Second World War.
Yeah, and this is exactly takes me back to the wounds. Once the fungus starts germinating, it's producing patcholine to knock out all the antagonists that they are colonizing the wounds, and this will facilitate its infection. Yeah.
So what's the meaning of these toxins? Why are they there? And especially on Alternaria, they are extra chromosomal. So
Yeah.
so
Maybe this is,
how are they?
yeah, this is an ecological fitness.
Exactly.
You know, all these alternaria that all of a sudden they are causing a disease. You know, maybe it's some other component that we don't know about them.
Missing link.
Yeah, yeah. Of course.
Well, I think, yes, it's quite late tonight because we had almost 12 hours meeting today. So our brain is at the end. But
Yeah.
I think it was a nice discussion, not with answers, because it was not supposed to give answers, but just to give ideas for thinking, for collaboration, for trying to find maybe in the future some solution to post-harvest issues, because there are more and more post-harvest issues coming out, and probably in every country you can realize that new problems that were not a problem are coming out, and it's really relevant to understand this, and the tool of the pathobiome can help into giving answers.
Yeah, this is, you know, the purpose was just to give an approach of thinking out of the box. We all like to be in our box, not going out and thinking out of the box about some other things that are there. So this was the main idea of this roundtable discussion, and I think it was very interesting, and I hope so it will promote some thinking for the future. And thank you all for your participation.